Researchers from the University of Cambridge have developed model embryos from mouse stem cells that form a brain, a beating heart, and the foundations of all the other organs of the body, a new avenue for recreating the first stages of life. The team developed the embryo model without eggs or sperm and instead used the stem cells, the body's master cells, which can develop into almost any cell type in the body. The researchers mimicked natural processes in the lab by guiding the three types of stem cells found in early mammalian development to the point where they start interacting. By inducing the expression of a particular set of genes and establishing a unique environment for their interactions, the researchers were able to get the stem cells to talk to each other. The stem cells self-organized into structures that progressed through the successive developmental stages until they had beating hearts and the foundations of the brain, as well as the yolk sac where the embryo develops and gets nutrients from in its first weeks. Unlike other synthetic embryos, the Cambridge developed models reached the point where the entire brain, including the anterior portion, began to develop. The team say their results, the result of more than a decade of research that progressively led to more and more complex embryo-like structures and reported in the journal Nature, could help researchers understand why some embryos fail while others go on to develop into a healthy pregnancy. Additionally, the results could be used to guide repair and development of synthetic human organs for transplantation. The researcher says that their mouse embryo model not only develops a brain but also a beating heart, all the components that go on to make up the body. For a human embryo to develop successfully, there needs to be a dialogue between the tissues that will become the embryo and the tissues that will connect the embryo to the mother. In the first week after fertilization, three types of stem cells develop. One will eventually become the tissues of the body and the other to support the embryo's development. One of these extra embryonic stem cell types will become the placenta, which connects the fetus to the mother and provides oxygen and nutrients. And the second is the yolk sac, where the embryo grows and where it gets its nutrients from in early development. Many pregnancies fail at the point when the three types of stem cells begin to send mechanical and chemical signals to each other, which tell the embryo how to develop properly. Over the past decade, the research group has been studying these earliest stages of pregnancy in order to understand why some pregnancies fail and some succeed. To guide the development of their synthetic embryo, the researchers put together cultured stem cells representing each of the three types of tissue in the right proportions and environment to promote their growth and communication with each other, eventually self-assembling into an embryo. A major advance in the study is the ability to generate the entire brain, in particular the anterior part, which has been a major goal in the development of synthetic embryos. While the current research was carried out in mouse models, the researchers are developing similar human models with the potential to be directed towards the generation of specific organ types to understand mechanisms behind crucial processes that would be otherwise impossible to study in real embryos. At present, UK law permits human embryos to be studied in the laboratory only up to the 14th day of development. If these new methods are shown to be successful with human stem cells in future, they could also be used to guide development of synthetic organs for patients awaiting transplants. There are so many people around the world who wait for years for organ transplants. What makes this work so exciting is that the knowledge coming out of it could be used to grow correct synthetic human organs to save lives that are currently lost. It should also be possible to affect and heal adult organs by using the knowledge the scientists have on how they are made.